This PowerPoint will examine hemolytic anemia and heme degradation in the context of hereditary spherocytosis. Marcus Dudley, a 2.5-year-old African American male, presents with the chief complaint of fatigue and jaundice. His birth was normal and his family history is unremarkable. His eyes showed mild scleroicterus, consistent with laboratory values of elevated bilirubin. His hematocrit and hemoglobin was decreased, indicative of anemia, which is causing the fatigue. Further laboratory reports demonstrate the presence of spherocytes in a blood smear and increased osmotic fragility of the red blood cell from a blood sample. He was diagnosed with hereditary spherocytosis. Hereditary spherocytosis arises from a mutation in the spectrin cytoskeleton, or the proteins like ankyrin and BAN3 that attach the cytoskeleton to the membrane. Many of the red blood cells in a blood smear will have a spherical shape when examined under a light microscope. Remember that the sphere is the minimal surface area that can hold the cellular contents, so that this has an effect of increasing the hemoglobin concentration. This means that patients with hereditary spherocytosis are often will have red blood cells that are hyperchromatic. The spherical red blood cells are fragile and non-deformable. Therefore, the spherocytes cannot easily traverse the spleen, resulting in their removal and degradation by macrophages. This leads to large cell turnover and an increased production of bilirubin. This figure shows the general structure of the red blood cell surface looking from the intracellular point of view. The cytoskeleton is made from spectrin, a heterotetrameric protein that forms a coil structure that is shown in red. Note that the twisting spectrin structure does not penetrate the membrane, that is, spectrin is membrane associated and not a transmembrane protein. Spectrin is attached to the membrane by several other transmembrane proteins such as BAN3, which is shown as green cylinders, and glycophorins, which are shown in maroon. It's also associated with other membrane-associated proteins like ankyrin, which is shown in yellow, and BAM4.1, which is shown as light blue spheres, adductin, which is shown as light green, and actin, which is shown as purple spheres. Note that the BAN3 also functions as a transporter that exchanges bicarbonates and chloride ions. The membrane includes a number of other proteins, including the glucose transporter GLUT1. The top left figure shows the association of an alpha spectrin shown in red coil wrapping around a copy of the beta spectrum, which is shown in orange. A pair of these coil alpha-beta dimers associate to form the heterotetramer, which is shown at the bottom left panel. The right panel shows the raw data from an image made from electron microscopy. Spectrin is assembled to form the network that allows the red blood cell to deform in order to pass through the narrow capillaries. Mutations in spectrum result in the formation of spheres. In the case of hereditary spherocytosis, results in the formation of elliptical shapes, as in the case of hereditary elliptocytosis. The difference between a red blood cell having a spherical shape and a red blood cell that has elliptical shape is due to the location of the mutations. For example, mutations in ankyrin will reduce or eliminate the association of spectrum with the membrane. This set of mutations will produce spherical shaped cells. Mutations that map to the areas with red circles will cause spherocytosis. Mutations which inhibit two spectrin dimers from associating with each other will produce a red blood cell with elliptical shape. These mutations can be mapped to the structures within which are shown in the black circles. Patients with hereditary elliptocytosis will have similar symptoms as patients with hereditary spherocytosis. This is a slight difference in the shape of the cell. The red blood cell of patients with hereditary spherocytosis will show more lysis and hypotonic solutions when compared to normal patients. The osmotic fragility test involves a combination of blood with differing salt solutions that are varied between 0 and 1%. That is 1 gram of sodium chloride per 100 mils to 0 grams of sodium chloride per 100 mils. The sample is equilibrated and then spun down in a centrifuge to remove intact cells. Samples with increased hemolysis, for example, the bottom row, will have more red color than the wild-type controls, which are the top row. In this case, the increase of osmotic fragility is caused by a lack of a well-defined spectrin cytoskeleton. Hereditary spherocytosis is one of many disorders that causes rapid red blood cell turnover because of extravascular hemolysis. That is, the cells are removed by macrophages present in the spleen. There are some disorders that result in intravascular hemolysis, that is, hemolysis within the circulation system. Intravascular hemolysis is associated with symptoms caused by the presence of cell-free hemoglobin. An increased amount of intravascular and extravascular hemolysis will both produce elevated levels of the yellow-colored bilirubin due to the breakdown of the heme group. Elevated levels of bilirubin will cause jaundice, which is first seen as the yellowing of the sclera of the eyes and then seen as the yellowing of the skin as the bilirubin diffuses into the peripheral tissues. Jaundice is associated with diseases that result in higher than normal turnover of the red blood cells, or with individuals with problems with either the liver or bile duct. The first step of heme degradation results in the cleavage of the porphyrin ring by the addition of two molecules of oxygen. This reaction produces carbon monoxide, free ferric iron, and biliverdin as products and is catalyzed by heme oxygenase, a copper-containing enzyme. The second step involves the reduction of biliverdin to form bilirubin. The reducing equivalence from both reactions comes from NADPH. And remember that the primary source of NADPH is the initial steps of the pentose phosphate shunts, including glucose 6-phosphate dehydrogenase. Note that the carbon monoxide, or just remember, carbon monoxide binds to hemoglobin and reduces the effectiveness of oxygen transport, as seen in the figure to the right. This is because carbon monoxide acts as a competitive inhibitor with oxygen for its binding to hemoglobin, and it acts as a allosteric effector, shifting the equilibrium toward the R state and increasing the oxygen affinity of the hemoglobin. Heme breakdown takes place in the spleen when extravascular hemolysis is present, or by circulating macrophages when intravascular hemolysis is present. The hydrophobic bilirubin is secreted from the macrophages and is carried to the liver by serum albumin. The liver conjugates bilirubin to glucuronic acid, thereby increasing the water solubility of this intermediate. The conjugated bilirubin is normally secreted into the bile. However, the slow transport of bilirubin to the bile will oftentimes allow some of the conjugated bilirubin to flow back into the circulation system. A blockage of the bile duct will also cause an increased accumulation of conjugated bilirubin in the circulation system. Once the bilirubin is in the intestines, bacteria can convert it to urobilinogens and stercobilins or stercobilinogens, which are excreted from the body. Intravascular hemolysis is characterized by jaundice, hypertension, and tissue damage. The jaundice is caused by an increase in heme breakdown as a result of increased red blood cell turnover. The hypertension is caused by an increase in vasoconstriction, which is in turn caused by a decrease in the concentration of the secondary messenger nitric oxide, a small molecule that is converted to nitrates by cell-free hemoglobin. Tissue damage is caused by reactive oxygen species such as superoxide, which is generated by cell-free hemoglobin. The red blood cell effectively concentrates the hemoglobin within the intercellular compartment so that the hemoglobin remains a tetramer. Upon intravascular hemolysis, the hemoglobin is released into the circulation system and the water accessible area beyond the endothelial cells. This effectively reduces the concentration of hemoglobin by 1,000-fold or more, thereby promoting the dissociation of the tetramer into two alpha-beta dimers. The 32 kilodalton dimer will be more prone to leave the circulation system, be more prone to not be filtered by the kidneys, and be more prone to generate reactive oxygen species. The red blood cell protects the hemoglobin from extracellular nitric oxide by preventing this small molecule from getting into the red blood cell and reacting with the hemoglobin. However, any hemoglobin that is released into the circulation system will be more likely to react with nitric oxide and will also be more likely to produce reactive oxygen species that will damage the tissue. This figure shows some of the important reactions. Nitric oxide is made from arginine by nitric oxide synthase, and nitric oxide functions as a secondary messenger that mediates vasodilation. Nitric oxide will bind to unligated heme groups, forming a covalent bond in the same way as molecular oxygen. The nitric oxide will remain bound to the heme until it encounters molecular oxygen. The two diatomic molecules will react, forming nitrate ions. This reaction is catalyzed by the heme iron and results in the oxidation of the heme iron. Note the same reaction will also occur between nitric oxide and oxyhemoglobin. That is, when nitric oxide encounters oxygen bound to the heme group, it will react with the oxygen to form nitrates, and resulting in the oxidation of the heme iron. The net effect is that hemoglobin is oxidized and that nitric oxide levels is decreased. Decreased levels of nitric oxide leads to vasoconstriction, and long-term vasoconstriction will cause hypertension. Now to conclude, Marcus Dudley, a child presents with the chief complaint of anemia and jaundice caused by increased red blood cell turnover. Lab values demonstrate that he had normal cytic hyperchromatic anemia. Furthermore, the yellowing of the eyes and increased serum bilirubin, both indirect and direct, indicate jaundice, and the presence of the spherocytes in the blood smear and the decreased osmotic fragility of blood samples confirm the presence of red